All right, welcome back. Here we are starting our class and back from spring break. And we have our first exam on Wednesday. And so we want to focus on that some today. If students have questions and so forth, we can keep answering those. But if we're set, I'll move on to the new material that will be after the first exam. And so I just want to show you this again. I think I showed this before break. If you're around next school year, this is a great internship program. I also wanted to show you this new. It's here in Portland, and it's sponsored by this Bioscience Association of Maine organization, and they generally have good food, like hors d'oeuvre type food at their events. So I would go to this if I were a student. And it's free, and you can get the link off of our recording like that, okay? So as always, we gotta work on our capstone journal. Just to think about that, right? We're near the end of March, and then April will be, the last month of the semester. So we want to shift into the mentality that, after our exam, we want to be learning new material and also really emphasizing working toward our capstone project effort, like that, working in your journal and starting to do some writing. And we've got our midterm on Wednesday that covers the first half of our course. So you'll see that if we start talking about new material today, it'll actually be that second half of our course and topics. So in one sense, if your capstone project fits into the topics in our first half of our course, after our first exam, you'll essentially be ready to really, work on your capstone project. On the other hand, the second half of our course, you'll be adding more stuff to it like that and learning those concepts and principles. And I think either of those situations is a good one to be in. And then we do have a week from Wednesday, this second capstone writing project, and I posted a description of that. And it's a challenging one to think about. Here's something you can think about it with regard to this, and that is, I'm trying hard to help students see that you can use AI not to write stuff. That's the big anxiety in education is the students are just going to have the AI write their paper, and you can't do that. You can't have AI write your paper or write sections or paragraphs on your paper. But in my experience, AI is just great for research and pulling stuff together. And that includes trying to understand the second writing assignment, not to produce the product, but to find out what would be ideas, right? Because it's about ethics, and you may have had a general education course associated with ethics, maybe more or less. But ethics can come at this topic from a variety of different angles. I made a little video to try to help you with that and try to grow in that direction, because as part of the capstone, as part of general education, we want to be reflective on how we fit in ethically, right? The state paid for half your education. It's a public university, and they paid for quite half, but the state's paid a lot. So what's essentially your social responsibility, and how do you feel and think about that, right? So the ethics is usually in biology courses, the limit is just like, is this ethical to do these experiments and to do this work and try to do CRISPR on a newborn baby. But there's a lot of different angles to consider this, and how you fit in, how our course fits in, how the topic fits in. And you're welcome to use AI to get some hints and maybe some guidance toward what up, like that, and what would the topics be? That's great. Put it in your journal, right? Because I'm super happy to see you working on your journal, and it includes if you're working on your journal with AI. I was thinking over break, and I was thinking that, because faculty talk about AI and try to talk about how to, you should just ban it or use it, and we should be using it, and one way to think about it is just to back up in history and think of prior technology, dial it back to the invention of books, and whether, there were folks before books that would memorize the cultural stories like that, right? And they're like, well, that's a ripoff, that's a low, people aren't even going to be able to think, right? Because books will just take their thinking away, right? So books are a good tool, and then there's more modern resources than that. Go on and on and on. The library is a good tool, like that. Well, so AI is a good tool, and in a sense, I think that we might be, one of the more desirable generations because it shifts us up to be able to really use to use our brain to take advantage of the tools of AI, right? It's not that AI is totally overwhelming us and way ahead of us. We could still harness it and figure out how to use it, take advantage of it. And this would be a good example of it, and more broadly, your overall project is a good one, right? I think, getting it, using it as a partner, and then showing, that that's a source and helper like that, okay? So we got to put that together, and it's a challenge like that, so I try to get that assignment out there in a little video to try to help think about that. So we're rolling in those directions. And our biggest thing today will be preparing for our course, our exam. And so there's 2 parts to our course, and the exam is about this, and that's the second half of our course that starts after our exam. And we talked a little bit about this. This is a really significant aspect of this that really drives the field forward, but it's just a whole nother kind of thing to open up, because evolution is the study and understanding and principles and concepts and terms in evolution are a big thing. But we tried to pick some of those up, and I've said that if you can pick some of those up, like as individual students, right, you went after some of those when we were picking examples, and you have that knowledge, either directly from having done it or some from having heard about it, you can get some more of that, because it's good to fit that into either, of our exams, but especially our first exam on Wednesday, just even if it's not asked for, to put it in the context. It'll say, describe an example, and you can add to that, this idea of the role that this could play in evolution and speciation and stuff like that, right? So just on the exam, it's going to be essay type exam. You don't have to BS. You don't have to try to fluff it up, but you can put stuff in. You can even put in stuff that you've been learning from your capstone project. Just put it in if it fits. You can put it in together. That's absolutely fine. Okay, and so I just wanted to show you this because of this, and I just failed to, I was looking back and thinking about it, and here's something that I failed to talk about. So phenotypic plasticity was going to fall into these 2 types of phenotypic plasticity. Either a continuous gradation, that's called a reaction norm, or discrete differences, that's polyphenism. And so this is a good one like that, where in general, in animals, body parts scale with the overall mass of the organism. So in every species of plant and animal, that species has a size range. So there are smaller versions of the species and larger versions of the species. And in general, the smaller species have smaller individual body parts. So in general, you can draw a correlation between this, like human, say height, and then think of finger. It generally, I have long fingers because I'm tall. Like there's a scaling, and that's a reaction norm. It's a graded reaction norm, and that can be, due to a mixture of genetic heritability and also environmental factors, right? Like that. And then we can think of that as a reaction norm, and then we can think of it as a polyphenism in that specific story, like that. And the book and our course, we pick more polyphenisms because they're just more kind of accessible. A reaction norm just seemed kind of obvious. You're like, well, duh, taller people have longer arms, and kind of in loose correlation. Well, yeah, I hope so, like that, right? Like that. So just to kind of look back on some of that, classmate Mike sent me this, just share this, and it'll be on recording so you don't have to scramble to write it down. But Mike's had courses with me before where I've had longer answer questions, and I like to talk about them like it's a story. I think that's a good way to write an essay, is to think about it as a story, to explain it. And so, Mike says, think about it, try to think of your answers that you're preparing as, steps in kind of a logical sequence. And we've been talking about it. That's not new. It's just putting it together, because it is hard for, I think, science students to put together a story, you know, like a longer answer, right? You're really used to seeing, like, you know, what's the site of protein synthesis? Oh, ribosome, and move on to the next question. So I think that's a helpful resource, and like I said, it'll be there. I'll try to remember to also add it to a Brightspace announcement too, so you can see that, you know, as part of our recording today. Yep. And so, you know, here's a good example of that, right? We have this big picture image like this, and you are welcome to draw images like that and label simple images like that on your exam answers, because image is often worth 1,000 words. But you also have to explain. You can't just use images because there's no real evidence that you understand, you know, the concepts that are displayed there, right? But you can do that, and certainly in your preparation, you can think of it, because that goes back to that concept map we talked about briefly, you know, where you can organize your thinking and your project and all your learning into, like, you know, kind of lay it out on a table and say, oh, over here is where I want to put that, and over here. And then you can build up more information. So that's a good thing. And you can see on here, for example, that this was always up. I showed this before, but right, we added to it. There's the second half of our last assigned reading is more leaning toward cancer biology and the diseases of old age associated with the life cycle, like that, right? All right. So I just made a couple of slides like this, right? So here's a take-home concept, right? So I think that that's a really good thing to always be thinking about as a biology, you know, person is like, well, what's the take-home concept here? What's the bottom line? Because I want to have a solid foundation to build stuff up on, you know. I don't want to just memorize a list of terms or a stack of flashcards. I want to have a thing that I would be like, here's the first thing I want to say, and it's the general kind of concept. So I wrote that out, and again, you know, I write stuff out on slides and I don't slow down, because hopefully the recording works and you'll go back and be able to write them down and study from that. And so I just try to create that. I'll read through it. So studying phenotypic plasticity, sometimes in model organisms and sometimes in non-model organisms. So that's a concept that is really good to try to get into our exam, at least in one place. That's the key thing of our field, is that these scientists have been studying model organisms under controlled environments in labs, and they've just learned so much that on any nature walk, if you come up to a mushroom and say, oh, look at how different those mushrooms look, and then there's like, oh, they're the same species, you'll be like, well, I bet we could get some biologists to suggest what the mechanisms might be and what genes might be involved and pathways and molecules might be involved in this story. It's just remarkable. And it rooted in evolution, rooted in the relationship between all these species. And it's just it's dramatic, right? How valuable, how significant that is. And so, you know, that's really helpful. And, you know, in some of our examples, it is a model organism. So the nematode is a model organism for studying lifespan. Then they turn to other non-model organisms like naked mole rats and so forth. And so then those molecular and cellular mechanisms, they become valuable and significant to help illuminate the way that these organisms fit into their environment. So we have an example like nematode, and that's our first example. So I just wanted to mention that example because we had the reading about that, where it's like the first half is about lifespan, and then the second half is about examples of older organisms and issues that could be associated with their demise, like that, okay? So that's an example. And I've got a couple of slides really kind of as a review, but just to kind of remind you of that, you know, like that, just to show you that. You know, we talked about these topics, and you want to know those to fit those in, and you want to fit them in that setting, like that, okay? Then this second example, I want to talk a little bit more about, because I realized in looking back over stuff that we talked about this idea of quorum sensing, and I just want to talk about that a little bit more. And so it occurs in this bobtail squid, and this quorum sensing occurs not in the squid, but in the bacteria that are symbiotic bacteria that live in its light organ. And it provides an example of a polyphenism, and the polyphenism is in the squid, whether it makes that light organ for the bacteria to live in and support them, or it doesn't, because the bacteria are either there or not in the environment. And the bacteria genus is Vibrio, and they're present already in the environment, and they can be pulled into the light organ as it develops. And if they're not in the environment, it doesn't develop. So that's the polyphenism based on the environment, like that. And we really like to focus on the organism, the metazoan, the animal or the plant, not so much the single-cell bacteria. But in talking about this in our course, we have mentioned a couple of times about bacteria and how cellular, molecular, genetic mechanisms in bacteria really provided, it's pretty clear they provided the evolutionary history to set the stage for multicellularity, and multicellularity that has this phenotypic plasticity. So we did want to talk about that some, and the term is called quorum sensing, and I just have a slide of that. And what it means is that every cell in the environment is making a molecule that's that triangle detector molecule, and that means that all the concentration in the environment is based on the number of the cells in that local environment. So it's a way of measuring the density of the environment by each cell, because each cell produces it, but also has a receptor for it. And if they're on their own, they don't detect much. But as soon as they start getting neighbors, they start detecting the overall concentration, the overall molarity of that autoinducer in the environment. And the cell is built to respond to that, and at a certain threshold concentration, it changes. It makes a change, and that's bacteria. That's bacteria making a predicted adaptive response based on the environment, and the environment is the overall number of the species in the neighborhood. Like that, right? And that's quorum sensing, and it's a different story, but in the nematode decides to extend its lifespan because of a variety of possible factors in the environment. So one is just nutrients. So if it's poor nutrients, it will slow down its rate of growth. Another is temperature. If it's a high temperature, it'll slow down its maturation because it's stressed and it doesn't want to try to reproduce under a stressful thing. And the third is what we mentioned, and that is the density. And so this is the bacteria in the squid, and lots of bacteria can do this, not all bacteria, but they can do this. And the nematode is doing that same thing, where each nematode is making a chemical, it's called a pheromone in that case, and measuring the concentration of it, and measuring therefore the density of its species in the environment. And at high densities, they'll go into diapause and hope that somehow the density will decrease over time, like that. Yep. So nice image of that from initially bacteria, and just that mechanism is just that straightforward. It's a kind of a curious thing. I'll just mention this off to the side. So it turns out that because if you look in an introductory biology textbook, there'll be a figure in every introductory biology textbook, and it will show this. It'll show, oh, here's a concept from the field of endocrinology, the study of hormones. And the first panel will show a hormone diffusing throughout the whole organism. And it says hormones are called endocrine factors and are the basis for the entire field of endocrinology. And the student learns about endocrine factors in that first semester of biology. But the biology textbook, even at that introductory level, wants to also show a second panel, and it says there are other molecules that just don't go through the whole organism. They're local factors, and they're called paracrine factors. And that's a big deal because it's very significant to have, for example, the skin cells underneath the scab heal, but we don't need to try to heal all the skin cells in our body. So we want a local signaling factor in many, many ways at a time. The third panel is the curious one because it's always there, and you're like, why is that there? And so the third panel shows this. It shows a cell making a molecule that it releases to the extracellular environment and then responding to it, and that's called autocrine. And I remember seeing it, way back when I was in college and going, what is that there for? Because an autocrine response is always either an endocrine response or a paracrine response. And it was just never clear to me why that would, why bother with that? But the answer is the textbook authors, they don't know why they included it either. It's endocrinologists that told them to do it. And it's because there are many endocrine glands that also have autocrine feedback. Either positive feedback going up and increasing the hormone they're making, or negative feedback to turn it off. So it's very, very common in that field as a way of regulating. And so it's an old strategy, to put a signal out there and then see what's coming back like that. What's the overall concentration? So it's a good example. And again, we want to be like, and it's not, that's not developmental biology, right? It's just a bacteria. But you set the stage for what the nematode does in its response environment, and some other organisms do that too, like that. Yep. And so very cool. So mechanism like that. And it was discovered initially by scientists growing the bacteria that was from that squid-like organ, and now it's seen in a variety of bacteria in a variety of different settings like that. So then I mentioned that there's that second reason, and that is that, what is the second reason? Oh, just the first reason is it exists, the quorum sensing exists in bacteria. The second reason is that it's a precedent for evolution of multicellularity. And then the third is what I just described. It's actually used in multicellular organisms for that purpose. Yep. So it's a good one like that to be able to get a good handle on that in the bobtail squid like that. Okay. All right. So then just pause there because I have some more stuff to say, but I kind of wanted to get through that and then stop and see about how you're doing and if you have questions about anything at all about the exam. Yep.
You had a slide a couple weeks ago that was like an overview of kind of everything that we've covered, if I remember correctly, and I couldn't find that when I was going back to the videos.
Oh, okay.
Is there?
Yeah. Yep. Yeah. So I made up that. I was like, I think I know what you mean, and I think it was like, I tried to like make a little outline of chapter 1.
Yeah.
Yeah. So I could add that. Like I can add that Mike's image, answer template and an outline of chapter 1. And I did that quickly just to kind of help, not because I was like, I was just trying to guide that toward, saying you can make an outline, or you can make a concept map to try to build it out like that. But yeah, I put up an outline of chapter 1 and chapter 2, just, to kind of help do that. Yep. Yeah. Yeah, I can put that on the Brightspace, like for our recording, I can add some attachments. Yep.
I also saw, like, during the video that you showed that you sent out, just going over, like, basically what we didn't get to go over.
Yeah. Yeah.
In my example, I was curious too, and I was glad that you went over it. But you basically said that the water, the Daphnia, are too small to have the ecdysteroid response. Can you like talk about what that kind of means?
Yeah, so I think that's just kind of my made up idea of that that it, and I don't even know if it's biologically true.
I was curious too because the paper does not mention it at all.
Yeah.
I was like, because that was one of the questions I wanted to ask you.
Yeah.
I was like, why does this not talk about this? But
yeah, because so in that case, in this little water flea Daphnia, it uses a mechanism of signaling that's this kind of electrical channel thing that they found evidence for. And it's curious, we have so much emphasis on hormones like that. And there are signaling molecules in all animals, but in really little animals, they have less reliance on hormones trying to coordinate it, and they have sometimes other non-hormonal mechanisms. So it was really just a brief comment like that, I think, rather than like opening a big door. Yeah.
Do those like signal? Is that signaling? Is that been researched in like to have a connection connection with ecdysteroid and like bigger?
Yeah, I don't know. That was a kind of an original paper where they were looking at all the gene expression, and they came up with this one and started looking into it. And that's the first time I had seen anybody doing that in Daphnia. Like our book has that other example where the Daphnia can make a helmet instead of those neck teeth like that. And I guess there are some other kind of morphological changes that the Daphnia can make. And I just had never seen articles where they were diving into that. But it's a great article because people can do that, right? They can dive in and if they have like the Daphnia genome sequence, they can just dive in and say, well, what changes in gene expression do we see? And what are those genes? Yep.
For this course, though, probably not a super valuable example for like something like the exam, just because there's not a lot of formula.
Well, I think it's a really good example because it's a really good example of transgenerational changes that can occur, and those exist. But it seems to me we didn't really dive into this, but the kind of the most striking examples of those are in some pretty simple organisms. It's just kind of a quick example of why it's a little bit complicated. So for example, in mammals, people like to talk about, I think that's a transgenerational change of phenotypic plasticity based on the pregnant mom's experience, her environment. But you can't study that first generation because they're already present, right? So the fetus or embryo is already there, and so it's not really fair to say, that's transgenerational because the environment that was affecting mom could have also just been directly affecting the embryo or fetus like that. And then people will point out that in the embryo or fetus of a mammal, they already have the germline cells, the cells that are going to make sperm and egg for the next generation. So there are a lot of people that say, well, I'm really not convinced with transgenerational examples in mammals unless it's the fourth generation, because 3 generations are already, immediately present, potentially experiencing the environment. So that reason and others makes it so that sometimes these simple organisms, like I see that, right, like that. And they also, they have such short generation times that you can see that transgenerational effect is inherited, and then if you remove it, right, it's predictive adaptive response. If you remove it, the Daphnia will be like that generation, and it'll be somewhat like that the next generation, but it will eventually go back to not having neck teeth or a helmet like that. So, really satisfying examples of transgenerational polyphenism like that. Yeah, so I think that was, they're good examples like in our book or those. And so it was cool that they started looking at, well, what genes are affected here, because what's the mechanism like that? And I thought that was interesting, but I just don't know of anybody else that's going after that. And I'm sure people have, I just don't know it. Yeah. Yep. Yeah. Anything others people want to talk about? Yeah. All right. So let me say, let's see what we're going to talk about here. So we're going to keep talking about stuff, not necessarily to intentionally try to review, because you're doing that, you're studying, but to kind of open the door for questions and stuff like that, and also try to coach you and help you, because I think it's a big thing to ask somebody to write an essay exam. So here's a thing that I'm going to do that will be helpful, okay? And that is, we're going to keep working in class, get ready, and asking questions and stuff. But Brightspace, what I'm going to do today is I will post the exam. And so you can't bring any notes, but you'll be able to study and focus and prepare your examples. So the exam is going to have, it has 3 questions that are 20 points each, and it's like, each question is a little bit long, because it's like, give an example of this, describe it, and then it has 4 questions that are 10 point each, like that. I'm just going to send it out. I'm not going to do that for the final, because when somebody does that, the students immediately stop learning about the rest of the course and they're just gonna focus on preparing their examples. So that's why I haven't done it in other classes. But I think it's just, I'm very sensitive. There's this thing in pedagogy that's called scaffold or step or sequence learning. And that is, well, hey, I want you to learn how to jump up onto the top of this building. And you're like, I guess everybody's gonna fail, So you have to build some steps like that. So the idea is build a step to help like that. So again, can't bring notes like that. And it'd be sad, if you just tried to have AI write it at home and then you memorize the answer from AI, right? And I tried to write questions that are not really amenable to just AI going like that. But yeah, it's gonna have to be you writing, right? So it's the same exam, but it's your study guide. It's a pretty good study guide like that, yeah? And you'll see, it's a lot of information. So that's true too, that there's a lot of stuff to write down. And again, don't BS, but bring what you got, and the questions are trying to be, both specific and open. So put stuff in, that you learned about too, and prepare your answers. That's a good thing, okay? So I'll do that today, and then you'll see that exam. And you have to answer there are 7 questions, you have to answer all of them like that, yep. So just talk about that, because then, we'll keep talking, because I included this, right? A little kind of outline, right? Like this, anytime we talk about a metazoan is just a multicellular plant or animal, so plant or animal symbiosis with a microbe, and so, like Mike said, it's a story, so let's try to start piecing together outlines of stories, just to show you that framework, that strategy, right? Like that. Those are good things to be able to say, to explain the story, right? And that's, that's an acquired skill, right? Practice skill, right? To say, oh, that bobtail squid, what can you tell me about it? And you're like, well, I should organize it into, a story, a series of, organized kind of paragraphs of like, well, first I want to tell you about what's up with that, yep. And so just made that to kind of try to help with that idea. And then, I made this, from the book, in our reading, right? The book dives into it in some great detail, and you're not expected to learn those great details on the exam, but you could get the framework of the story from that. And in all the courses I teach, I say that's the challenge with biology, is there's too much information. And so instead of walking away with none, like I'm overwhelmed, I got nothing out of that I could repeat. Try to get the pieces, and build up the pieces. What's the take-home message here and there, and like that, right? Okay, so just some of that. And doing that, I also like to show visuals, so I don't talk through this, just to show you this. And this is, originally, I show this part of, it's an image I made for developmental biology. So this is the case with all developmental stories on Earth, where we start with a fertilized egg, and we're going to make a mature multicellular organism, plant or animal. So it's a universal one. And there's going to be cell proliferation, it's multicellular. And then there's going to be growth and maturation. And that's a timeline. So in addition to showing you this, I like to try to think about how could we look at something like that and make anything. So squid, tree, whatever it is. So I usually talk about animals. It seems like everyone except Emily is focused on animals for their project like that, rather than plants. And so we think of like early development and then later development. So embryonic development, post-embryonic development. In some cases that could be, for animals, that could be prior to release from the mom body, or it's all happening out after fertilization takes place outside of the parents' bodies, like that, right? So this was early, this was embryology, but now developmental biologists study the whole life cycle, and we do too. So right, lots of parts of our course are that later maturation, like that. And it's just really interesting to see here. Like over break, I listened to a professor talking about studying, oh gosh, he studies the model organism zebrafish. So that's a vertebrate, it's a fish. You can just buy it in any aquarium pet store for sells tropical fish, just a little striped minnow. And they've done so much genetics on that. And it turns out that bony fish there are very many species of bony fish. Like half of all vertebrates are bony fish, and they have a lot of different shapes and body, morphology and structures evolved. And so these scientists they just sequence all the genomes, and they do this thing called phylomapping now, which is to take take a species and sequence the genome of everybody in this room, and then use those genomes to determine what are the variations within a human genome and what is the common universal human genome, and establish this common fixed genome for several species of closely related fish, and get a common genome for that group of fish and start comparing it with more distant genomes. And they're really focused on that maturation, because there's a lot of really significant development that happens in vertebrates after birth, and just the formation of the embryo is pretty complicated. But after birth, there's a lot of stories of maturation, and so it's just a huge area of research. And again, it's developmental biology. Our course fits into that. So there's a lot of that, right? Our topics involve a lot of that, right? Some transgenerational stuff happening, throughout the whole life cycle, but a lot of stuff about, reproductive maturation, sexual determination and so forth, aging, and that, like that, right? So the, example like that is from that stage of life, right? Like that, okay? And development always involves growth and patterning is the cells becoming different, and that's what the two things are that are happening. And so that's what's going to be happening in the topics we learn about. And there are really just three types of growth: proliferation, increasing numbers; cell enlargement, or releasing materials like bone, where there are a few cells in a bone, but the bone's pretty big because it's released that bony matrix extracellular material like that, right? And then the patterning means the cells becoming different, so we get different tissues like that. And this is the developmental plasticity is going to be at the level of some of these, at either the organ level or down at the individual, cells making little organs like a hair follicle type thing, like that, right? And so there's only two ways the cells become different, and it's either through unequal cell division or cell communication. And we've been talking a lot about cell communication, really endocrine factors, paracrine factors, or extracellular environmental signals, and it's just the center of how organisms grow and change. And developmental biologists call that cell communication that's been done by endocrine factors or paracrine factors, which developmental biologists call signaling factors, and that includes hormones, paracrine factors, neurotransmitters, and so forth, like that. Yep. And here's the difference here in our course is we just said, what if there's an external thing like another organism, and it's going to impinge on that developmental pathway and cause phenotypic plasticity by bringing signals in from outside of the organism, right? The organism really set up, these cells are really set up to be listening to the extracellular environment within the organism. And all we're doing is having them also respond to an extra organismal signal. And that really often leads a targeted cell or cells to make a response that then is communicated with others in the organism. Just really classic developmental biology, and I just was trying to make images, visuals of it like that to talk about. So it could be a living organism, like with the squid and the little bacteria, but we also have lots of examples of the non-living organisms: sunlight, diet, temperature, and on and on. Same thing. And again, here the vision is there's going to be some target molecules, probably not the whole organism, that are going to receive that signal, and could be those examples, and then communicate. And in that cell communication, a really common thing is there will be these set of signaling molecules, and there are really many of them that are very familiar to biologists now. You know, whether they're hormones, yeah, sure, you bet. We know a lot about insulin, or these local signaling factors, these paradigm factors. And in development, when there's a signal, it's very, very common for gene expression to change. So a lot of times, you know, we'll be talking about, well, what are the signaling molecules, what are those pathways, and then what's the target genes that are going to get regulated by a transcription factor. So just to give you some visuals of, that are in addition to, you know, lists and outlines, because you can draw this out, you can visualize it out, you can make a concept map that can help like that, okay? So I just did that and put that in, and then this is really moving on to the next material. So beyond our first exam. So we'll do that if we have time, but we're not in any hurry, because, and we shouldn't be discouraged because you're just like gonna wait till you see what's on the exam later today. But you know, we should be trying to spend time in here, because after I send out the exam, I'm not gonna like help answer questions like, well, what about the third question? Because I'm confused about that, you know, like that. And you're like, could you be my AI kind of thing? You know, I mean, you can still ask me questions, but if it's like that, which is what I did all the way through college, I'd just be like, I'm looking at question 1, and I'm wondering, you know, how you would answer this, or the kind of answer you'd like to see. Oh God, it's just embarrassing. But yeah, Bryce, did you have something? Or no, I thought.
Yeah, I was just when we were talking about cell communication there,
Yeah.
when I was reading our latest reading, the one where you're talking about cancer.
Yeah.
I was kind of confused when we were talking about the what is it right here, the cancer caused by miscommunication between cells, and the connection of that with the mesenchymal stromal cells.
Yeah. Yeah. So what's going on there is the author of our textbook really likes that idea, and it's an unusual idea, but it's relevant for our course, so we wanted to describe it. And the way to get there is to start out with like how everybody, scientists and the general public, doctors, how they understand what's the cause of cancer. And the general understanding is called the somatic mutation hypothesis, and that is cancer is caused by mutations, and it's the cancerous cells that have gotten mutated. They're the problem, and their problem is mutations, like that, you know? And so we should start out just with that before we get to what you're asking about, and talk some about that idea, you know, like that. But it goes with the ideas of like, maybe we could do surgery and remove those bad cells. They have mutations like that. Maybe we could monitor, you know, like after a person's been treated for cancer, if we can identify the genes that were mutated, we can try to determine if there are still some of those around, or if they come back like that. And so that's a really big, it's a model, and it's like, a lot of evidence for it, you know, and stuff like that is not wrong, but it's a way of understanding it, is that, you know, we got a lot of normal cells, and then, oops, mutations cause some cells directly to grow out of control, like that, yeah? And there is really, it's just a couple of scientific groups that push this other idea, and they're not discrediting, you know, what everybody else says. They're just trying to bring up the idea that it might not be the cells that grow out of control might not be the ones that are really having a problem, whether it's a mutation or some kind of environmental cause that's disturbing their normal function that leads them to affect how they interact with their neighboring cells. And it's the neighboring cells that are growing out of control, even though they're still normal cells. They're the cancer cells, but they're still normal, and they're receiving bad signals from other cells. And our author likes that in general, but our author likes that in this context because this chapter is late in the textbook, and the author is trying, you know, okay, we got the basics here, and let's take these kind of conceptually up a notch. And what our author really likes is what I was trying to show some in that, those drawings, and that is that when we talk about this course, ecological developmental plasticity, that we start out going, what we're talking about. We're talking about the environment, and we mean the environment that's outside of the organism, the extra-organismal environment, the sun, diet, stuff like that, right? And how is that affecting development, like that? And the author likes to think about that and include the idea that, well, multicellular organisms are still, the fundamental unit is still the cell. And so anytime you're talking about life, you're talking about a cell. And so a cell, it sees the external environment, and that includes the internal environment, so the signaling factors and all the molecules inside the organism, and also could be affected by the actual environment outside the organism. And so this is an example, and it gets complicated, right, confusing, but the author's really trying to get into that, get into that idea of phenotypic plasticity of the cell, and not really, because that's the real target, and that's the real effect, and it's not really the organism, it's these cells that are responding. And so he likes this example like that, and so that's the idea. And there's evidence for it, like, for example, there's some tissue settings, and one of them that's famous is in lungs. And so, for example, there's evidence that this very bad carcinogen asbestos doesn't cause mutations in the cells that are the lung cancer cells that grow, but instead the asbestos is causing disrupted normal tissue environment in the lung, and that leads to these changes, like that, yeah? And our author really pushes that idea forward. So that's what's going on there. When he's talking about stromal and mesenchymal, he's talking about the idea of two types of cells that are collaborating to make tissue, because that's always the thing with organs. Organs are a set of populations of cells that are different, that are teaming up like that. And our author just really likes the idea of, whoa, what if that communication gets messed up? Yeah?
So, if what you're talking about is a type of the senescence-associated secretory phenotype, you know how when the cell goes into senescence and it's not dividing anymore, but it's still metabolically active, and then they're pumping out those pro-inflammatory cytokines and the metalloproteinases, how does those getting pumped out break down the communication between the cells? So there's tissue breakdown or tissue miscommunication, like you said.
Yeah. So it we should try to before I address that, let me just say back to what we were talking about. A lot of scientists go, that's absolutely true, that cancer is affected by the neighboring cells, like that. And that original somatic mutation hypothesis is absolutely true. So what's happening is our author in this textbook is trying to kind of tease those things apart to help you illustrate those ideas. And our author knows they're different pieces, and we really want to pay attention to both of those, but they're actually blended together. Okay. And so in doing that, our author does that, and the author's trying to talk about aging, and get into, well, what is aging and what's happening? And author in that reading brings up some topics that are like that, we'll get to that, Mike, and question, but are treated like different sections for that reason of I want to introduce this concept. And then those topics do kind of blend and overlap. But it's good as learners to start out and just be going, okay, I'm going to turn our attention to this and just kind of try to focus on that. Because yeah, what you guys like Mike are, that's kind of this combination of the senescent cell phenotype description the author does, and then this tissue organization field theory, because they're a combination. But let's just start out by talking about them separately like that. And so in the talk, I tried to bring up, what is the author trying to emphasize? And the first one the author likes to want to talk about is DNA methylation. And I tried to show you in that, help you guide through that DNA methylation is always just the same thing, which is that in the DNA molecule, some of the C's, the AGCT, some of the C's, the cytosines, can get a methyl group covalently added to them. And when that happens, it seems to always cause the transcription gene expression to be reduced. So it's a way of reducing gene expression. And it comes up in our course in a good way because it is on the pathway of phenotypic plasticity. So we could have a story like this in those ants where the ants are going to develop as a worker or a queen based on the environment. And the difference is whether or not the workers feed the queen larvae or the worker larvae nutrients that have added this that are called royal jelly, which is fed to the queen, and it has a hormone in it. So that's going to be the environmental factor in this case. Is you could say it's the nutrients, the diet, because it's what's being fed to the larvae that are going to form queen or worker. And so that's the external signal. And it's a hormone, and it goes in and it's an unknown pathway, and it's going to affect changing gene expression. And then the changing gene expression is going to lead to changes in DNA methylation. And that's going to cause more changes in gene expression that are going to lead to the difference in the growth of either a queen or a worker. And so that's true in our course, that DNA methylation is often on the pathway, the steps within the organism to have that phenotypic plasticity. And that's probably because it does a really good job switching gene expression from one kind of set of genes to another. So that's the one, and we want to have that, be aware of that. You'll be able to use that in examples on our exam, in our capstone and so forth. In this chapter, then, the author is like, what is pretty interesting, that DNA methylation is pretty interesting because it has an additional role in aging. And that's a relatively new idea. And so I described it in the recording, which is that, for our bodies, our cells divide a lot. And so natural selection has favored the evolution of lots of DNA repair mechanisms because the DNA gets copied or it gets damaged and there's a mistake and could cause cancer, right? Somatic mutation hypothesis could cause cancer if it's in, a gene that causes cancer, like MYC or whatever. So there's really a high emphasis in general in plants and animals to repair DNA. Some animals that are really big and long-lived, like whales and elephants, have even higher DNA repair mechanisms than we do to try to clearly avoid that and reduce that aging, effect, because cancer is going to be an aging effect like that, okay? So it turns out that when you look at the S phase and the replication of DNA, the methylated Cs, the DNA methylation, that also gets replicated, gets copied as the cells grow and divide, copy. And it turns out that mechanism does not have very good accuracy. There's a lot of error in it. And so you can just use the pattern of DNA methylation and overall amount of DNA methylation, and you can just show it correlates with the age of the organism. that there are more differences. He compares identical twins. They're really similar when they're born. They get different. Part of that is because they have different real environments. Part of it is because the errors in the DNA methylation replication mechanism. And then he shows specific genes that pick up this DNA methylation. So that's one of the topics that he's like, I would like to dive into this and talk about it with regard to aging. And in that context, he brings up some examples. He's kind of doing a nature walk on this topic. And one of the examples he brings up that I talk about is this very unusual kind of cancer. It's a rare kind of cancer. It's a cancer of the gonads, either the testes or the ovaries. And they're called teratomas or teratocarcinomas or embryonic carcinomas. And in Maine, we're particularly interested in those because the pioneer of studying those types of cancers lived in Maine. And it's a guy named Leroy Hood back in the 1950s that worked up by Bar Harbor on Mount Desert Island at this Jackson Labs, and he studied mice. And he said those kind of cancers, and they're very, very curious cancers. And so we could talk some about his study from way back when. And but it was really pioneering and really super interesting. And for us, the key thing is this. If you take, we'll call them embryonic carcinoma cells. If you take one of those and you transplant it into the gonads of another mouse, they'll just get that cancer. It's a cancer cell, and it's got a problem with that cell. It's not the surrounding cells, it's that cell, and it'll just get that kind of cancer. And I said, did I say put it in the gonads? No, if you put it in the body, so you put it somewhere in the body, it'll get that kind of cancer. And so that, it's definitely got that problem. But if you take those cancer cells and that you put them in like a pre-implantation mouse embryo, then that cell will contribute to the embryo. And so in our developmental biology course, we learned about that idea that a pre-implantation mammalian embryo can accidentally split in two, and you can get identical twins. And you could do experimentally the reverse. You could take two different embryos and put them together, and you get one embryo that's made of a mixture of cells from both of the original embryos, and it grows up fine. So these very unusual embryonic carcinoma cells can be added to a normal pre-implantation embryo. They contribute to the formation of the embryo, and the embryo and then the animal that forms from it does not have that cancer. You can make those animals have their offspring be composed entirely from the original cells that were embryonic carcinoma cancer cells, and they no longer have that cancer, they no longer have a tendency to get that cancer. And scientists know really well what's going on. And what's going on is that in a really early mammalian embryo, there's a tremendous amount of removing DNA methylation. Because the early embryo cells are called embryonic stem cells. They can make any cell in the body. And so they're getting prepared to be able to express any gene. So let's remove not all the DNA methylation, it's very interesting, but lots of that. And that's what happens to the embryonic carcinoma. It's essentially like you erase the DNA methylation and you add them back. And the whole problem with the embryonic carcinoma is not a mutation, not an incorrect AGCT, but over time they had a mistake in the DNA methylation. And that just caused the wrong genes to express, like maybe they repressed a tumor suppressor gene like that, because it got methylated. So that can happen, and the author jumps into that example, and it's quite alarming to hear that, for your first time going, what? But that's what they're talking about. So lots of different pieces to try to add to DNA methylation to say, it seems like that's an issue with aging, concern with aging like that. So when we want to increase the lifespan and decrease the rate of aging, we're going to have to probably modulate that DNA methylation. So just to show you, that's a discrete example. It's included elsewhere in our course in normal examples and stuff like that, so that's frustrating. And then also, it's not the sole reason for aging, it's just one feature in aging. So then the author wants to move on to another situation with aging, and that's this senescent cell phenotype. and the idea of in a young organism, there are cellular interactions. Cells will be damaged or defective, and they get removed. Our immune system plays a big role in that, and so when there are damaged cells, a lot of times there can be inflammation to encourage our immune system to get in there and clean up the mess. And that is temporary inflammation, like when you twist your knee. And people call that sterile inflammation because you didn't get a cut, there's no bacteria, there's nothing to fight, off an infection. It's just that there's been tissue damage, and that has alerted the immune system to try to help take care of things. And the senescent cell phenotype is that becomes less and less effective as organisms get older. And so in an older organism, you see a lot of chronic sterile inflammation, and can include, the joints, like you might think, but also tissues like the heart and the lung and the liver. And those tissues start to have poor functioning. So that goes back to Mike's question. And so that's kind of, we should try, I think, keep that to stand alone, to say that just the local tissue environment gets to be a poorer quality in the older organism, and less quality functioning with regard to just keeping the cells growing and doing well. I don't think the author mentions it, but there's a really great example that you can look up, a fairly modern story, like maybe it's 20 years old now, where there is a procedure in biology that's called parabiosis, and it is where, let's see if we get this going. Let me just write this down. And again, just try to answer these in a way that, let's see. So it's this word is parabiosis, and I don't know, I don't think it's in our book, but it's just fun to hear about before you go off and have lunch and then stay for a exam. So parabiosis is where you just sew two living organisms together, and you're like, see how that works. So it's a very old school strategy. And these people at Stanford 20 years did that with mice, and they just sew together a really old mouse with a really young mouse, and the old mouse gets really young really fast, right? And one of the things it's doing is it's cleaning up that senescent cell phenotype really well, like that. And they've learned a lot about it. The people that did it initially were immunologists, and they learned that it's actually the older mouse's immune system that gets way better. And they found there's just a single protein, I think it's a molecule, single molecule, I think it's a protein in the young mouse's blood that can regenerate all the immune system function and a lot of just the regular cells function, and literally reverse the aging of the old mouse, like that. Yep. So that goes back to this role of the quality of the environment, the local environment. And remember, our author's loving that because our author loves to push you down the path of, oh, you thought it was just the environment outside the organism, but he likes the environment within the organism because the cell is experiencing that internal extracellular environment, like the tissue, like that, right? And I would like to just kind of try to keep that as a separate example from the tissue organization field theory. They fit together, and they have very overlapping details, but it's, I think, good just for us to just try to keep them separate as being like the senescent cell phenotype involves the immune system coming in and trying to help keep things looking good. The tissue organization field theory is really like with asbestos. When asbestos is in a lung, it is definitely causing chronic inflammation, chronic sterile inflammation, and also this idea that the cells, when they look at the cells that in an asbestos lung that are precancerous, the cells that are most disturbed in asbestos are not the cell type, like I'd say they are like the stromal cells that are disturbed by the presence of the asbestos particles, and it's actually the stromal cells that are the ones that have the lung cancer. And so, so long way to get around to try to talk about Mike's question, because I'd say yes, they're. I didn't remember exactly what you were asking, Mike, but they overlap. Those two stories overlap a lot. Senescent cell phenotype and that tissue organization field type. Yeah. But be able to say, I see this is one type of story and another. Both of them are cells and their neighborhood and the cellular neighborhood they're in. Yep.
Alright, so I have a follow-up question then.
Yeah.
Like, those are very overlapping, so that's, my question was kind of about that. But if we're gonna keep those two things separate, should we keep other things in our brain separate, like the royal jelly story? So that includes the DNA methyltransferases. Should we keep that story separate from, like, DNA methylation separate from FOXO, even though a lot of times they can involve each other, like, and tell two separate stories?
So if I'd say you always want to start to learn things separate, and then start putting them together. But it's really important to learn them separate so you can demonstrate. Yeah, I understand FOXO's transcription factor, and it can't get in its nucleus, can't do its job, like that. And then be like, and then of course DNA methylation also affects transcription, so that those can have these overlapping roles. The key thing I always worry about is when a student's learning and they start to get kind of the blur together thing going. Yeah, but if we want to try to connect them and bring them together, like the royal jelly story, which includes both that hormone and DNA methylation and gene expression and cell communication. Yep. Yeah. I don't know. It's just like I would think of it as like, here's a student and they learn how to find the mall. How do you get to the mall? So they instruct you to the mall, and then they are going to learn some more. And so the next thing to learn is how to get to Biddeford, and then how to get to Bangor. And then, on the exam, they're asked, well, how do you get to the mall? And they just have a mixture of answer, even though, because it just got too much, for a new person to learn and keep separate. So that's what we're trying to do. Yeah, but yeah, when possible, we definitely want to, include in our story, though, once we start talking about it, there are many of these features occur in a single story, like that aphid wing story. We looked at that flow chart and we're like, oh my gosh, we can look at different points of this flow chart and see, different good examples. That's just really good to be able to explain that, and keep them separate in your explanation, but yet, cover them, like, oh, I have 4 examples why that's a good story, like that.
So I have another example of that.
Yep.
Because I had written all of the bobtail squid stuff instead of using quorum sensin. I had put it as an outsourced queue like aphids, and I guess I should probably separate those two and talk about. Even though quorum sensing technically is an outsourced queue, it's a little bit different than quorum sensing. Outsourced queues include more things than just quorum sensing.
Yeah, and try to express your realization of that. You could say that literally on exam, because it's helpful, right? To demonstrate you appreciate that that there are these different perspectives and different steps involved, and we want to focus on one at a time, like that. Like our author did. I mean, I thought that reading was hard because the author is just kind of like just going across a whole bunch of territory, like kind of a grab bag of favorite topics. They're all good, but we want to be able to understand them one at a time, to be able to say, I can explain, why they brought up DNA methylation in an aging, chapter. Yep. Yeah. Other questions people have about anything at all?
So
Yeah.
maybe a
asbestos example, right? So is it like, is basically how it's affecting, when they're talking about cell miscommunication, like it's affecting the environment in between them? Like what's?
Yeah, and again, the way to think about it is, I would say is, well, there's normal cells, and then there are cancer cells. And so let's take a look at this cancer cell. And it's a cell, so it's the basic unit of life on Earth. And it's doing its thing. And are we labeling it a cancer cell because it's gotten changes and mutations in it that are permanent and defective, and we can point to them, and that's why it's a cancer cell? If we could, say, for example, use CRISPR and change the MYC mutation back to a wild type, it wouldn't be a cancer cell. So is it like that? Or is it that the cancer cell is surrounded by other cells that were normally supposed to help keep that cell in line and functioning normally, and have gotten disturbed and are giving them false, incorrect, inaccurate information? And I guess the author would say, yeah, because that second example is like a phenotypic plasticity story, where you could be a normal cell staying in line, or based on your external environment, extracellular environment, you could just get going down a different pathway. And that cancer itself could be an example of phenotypic plasticity when we look at the actual cell that's causing trouble and why it's causing trouble. Like that? Yeah. Yeah. And it's like some of this stuff is like, I have to sleep on that. It's like that. And so it's good, to learn, get exposed to that, but also that's not really what we're hoping to be able to express on the exam with great insight, right? This author he's a retired professor now, he's been thinking about this a lot longer than any of us, including me, have, so appreciate that, try to express it, be able to express that as best you can, but also recognize that it's a complicated concept, and it goes down to the point of the cell is the fundamental unit of life on Earth. And when you say, multicellular organism, you're saying those are individual cells that are working together, but they're still that, multicellular is a big surprise because of the idea of like a cell would go through apoptosis. Why would a cell do that? As far as the cell is concerned, it's all that matters. It's the basic unit of life. So there's features of multicellularity like that, and cancer, uncontrolled growth like that. How is it not common? Because cells are like, I'm on my own, I'm going for it, like that. Yep. And we're doing great. Other questions people have, we just have a couple more minutes. I kind of joked about not answering questions. I can still answer questions, email me questions, stuff like that. But again, it shouldn't be like, hey, can you just but it is good if there's like a logical typo on the exam too, where you're like, I think there's a typo kind of thing where it doesn't make sense, so let me know about that. Because when we come on Wednesday, we're just going to have the exam, you can't have any notes or anything like that, and you're just going to write your answers down, but you'll have seen the questions ahead of time. Yep, and that's not happening on the final. We're not going to do that in the final, because it is a thing where once you see it, you're like, that's all I'm ever going to learn, like that. Yep. Let's see, did you have another thing? Okay, so we can be good for now. Yep, and so I'll get the things posted, and I have a new computer, so we're hoping that we capture the recording, and it looks like we did.